Hi everyone, my name is Miora. I'm a PhD student from Madagascar. I'm working on the improvement of the mineral nutrition of rice, especially in phosphorus. I am pleased today to present you a part of our research entitled Mycorrhizal inoculation decreases phosphorus supply dependency of 4 rice varieties. We work released in Madagascar in collaboration with Professor Volatsara as a plant microbiologist, Dr. Jean Trapp as a soil functional ecologist, and Dr. Patrice AUTFRAY as an agronomist. Madagascar has the longest tradition of rice production. For the Malagasy people, rice is a core part of our diet. Many families eat rice 3 times a day. Well, in Madagascar, upland rice cultivation is constrained by soil acidity and the low bioavailability of phosphorus. In regard to their role in plant phosphorus nutrition, arbuscular mycorrhizal fungi could improve crop yield in poor nutrient soils. We wondered if there are varieties that interact with our mycorrhiza performing inoculated under low phosphorus input. 3 objectives were carried out for this study: to identify the efficiency levels of arbuscular mycorrhizal fungi for promoted rice cultivars, to test low-cost technologies concerning inoculant dose, to test low phosphorus supply in interaction with micromanure application. This study is the result of different contributions of multidisciplinary research on ecology, microbiology, and agronomy of plant soil fungus. And this biological symbiosis as ecological concept brings us to the development of agroecology, relationships between plants, fungus, and their environment, and could improve food security as in quantity and in quality. The field experiment completed 2 factors split plot design with 4 blocks as a repetition. Factor 1, variety with 4 upland varieties from national and international genetic bank. Fofifa 182, Fofifa 185, Nerica 4, and WAB. Factor 2, inoculation and phosphorus, or IP, with 4 modalities: without inoculation, without phosphorus, with inoculation without phosphorus, without inoculation with phosphorus, and with inoculation with phosphorus. In with inoculation, a commercial inoculant active from a Prometec company was used. This inoculant contained a Rhizophagus irregularis strain with 6,400 spores per gram. In with phosphorus, 20 kilos was applied using a triple superphosphate fertilizer. The pictures on the right show the different steps on the implementation of the experimentation. First one, the application of phosphorus in the pocket. The second one, the mixing of the seeds and the inoculum, which is done on the day of sowing. And the third one, to place the inoculated seeds in the pockets simultaneously with the manure to protect the inoculum from the sun. As a result, this slide shows the measured variables of the plants on 2 stages. At tillering, till number, shoot dry matter, phosphorus of the shoot, nitrogen of the shoot are measured. And at maturity, till number, straw dry matter, phosphorus of straw, nitrogen of the straw, grain yield, phosphorus of the grain, nitrogen of the grain are measured. Both stages showed the same results on both factors. With the rice varieties factor, there was no effect. The graphs on the right are therefore examples from the maturity stage, which represents the yield, the nitrogen uptaken from the grain, and the phosphorus uptaken from the grain on the y-axis, and the phosphorus inoculation treatments on the x-axis. With inoculation phosphorus factor, without phosphorus with inoculation is significantly higher than without phosphorus without inoculation, shown on the graphs circled in red. The next result is about the mycorrhizal parameters recorded at tilling. These pictures in left show the step of root sampling, root staining observed under microscope to evaluate the mycorrhization rate illustrated in the graph on y-axis in percent, and on the x-axis, global mycorrhizal frequency F, global mycorrhizal intensity M, and global arbuscular intensity A. Without phosphorus, no inoculation effect, but local and inoculated strains are present, marked with the green circle, and with phosphorus, there is inoculation effect, a significant decrease with inoculated strain. You can see with a red circle. Despite a high frequency, mycorrhizal intensity and arbuscular intensity are low. We can see with blue circle. The contributions to the Congress from this research is then to improve synergies of rice genetic and soil organisms, and to valorize promoted rice varieties for efficient arbuscular mycorrhizal fungi symbiosis, and to develop synergies between local and performant inoculant arbuscular mycorrhizal fungi. Thank you for your attention. Don't hesitate to ask questions.